kind introduction. It's nice to see you all here. Nice to be on this side of the podium. Okay, so today I'm going to talk about some work that we did in the last couple of years in the lab, and it all involves mitochondrial proteins and protein targeting. Oops, got the USB. Okay, so as some of you probably know, many of the proteins that start the way on ribosomes in the cytosol do not actually belong to the cytosol. And they have to be targeted and imported into different organelles in the cell. And this targeting and import is usually very efficient, but we know about more and more physiological and disease conditions that disrupt this process. And when I think about problems with protein importing into mitochondria, I think that it's kind of similar to a traffic jam. So imagine yourself standing in a traffic jam to your favorite national park. It has both direct and indirect consequences. Now, the direct consequences are quite clear. You're not hiking your favorite trail. You're stuck with your family in a car, for better and for worse. But this traffic jam might affect others. And these might be people that live nearby and trying to get to their homes after a long day of work. It can also be some wild animals that are trying to go to a different region of the park, which is actually something that I have witnessed in Yellowstone. This was taken from my car. So in the lab, many of the projects are focused on mitochondrial proteins. And the reason why we really care about mitochondria is because this organelle is just awesome. It's very important. So many of us think about it as the powerhouse of the cell because it produces ATP, the energy that is required for viability, but it's really a very small portion of what this organelle does. It can make many other metabolites that you can see here that are very essential for cell viability. And this is why I like to think about mitochondria as factories that manufacturing many metabolites that are essential for cells. In order to make this factory successful and running, we have to constantly import proteins that are basically the enzymes in these biosynthesis pathways and the factory workers into the organelle. Now, mitochondria, they do have a genome, but it's a very, very small one, and it encodes to only a handful of proteins, while 99% of the mitochondrial proteome is actually translated in the cytosol. And these proteins need to be targeted and imported into the mitochondria. This is how, in a very simplified manner, it happens. Mitochondrial proteins often have this barcode that you can see here in red that is called the MTS, the mitochondrial targeting signal. And this barcode is recognized by receptors on the surface of the organelle, and it's later going to be incorporated into this the translocases, TOM and TIM, and be pushed into the lumen of the organelle. When it reaches the organelle, the lumen, the targeting signal is no longer needed. So we cleave it, and we have the mature, folded, and functional protein. This is, as I said, a very simplified way to show you import. In reality, we have different compartments in the mitochondria, and we have different machineries that import proteins into these organelles. So we are talking about dozens of proteins and complexes that are involved in the protein of import, and also about other molecules like ATP and the maintenance of a membrane potential that are essential here. So it's not that surprising if I'll tell you that this is not always perfect. And we know that under physiological conditions like mitochondrial biogenesis cues, and in different diseases, we have problems with the import of proteins into the mitochondria, and that includes neurodegenerative diseases that are actually now thought to be more and more connected. So going back to the first slide analogy about the consequences of a traffic jam, let's think about what happens when proteins cannot reach the mitochondria. Again, the direct consequences are quite clear. We have an unfunctional factory. But we are now understanding more and more that the accumulation of these unimported proteins outside of the organelle is also something that we really don't want in the cell, and that can be very, very dangerous. These proteins can actually become a huge burden on the cytosolic protein quality control machinery and on the degradation machinery. And this is how people think that sometimes they affect the aggregation or the progression of the aggregation in diseases where we have, again, problems with these toxic aggregates like neurodegeneration. So from all these reasons, we care about how cells deal with it. How do they respond and how do they correct these problems with protein import? And today I'm going to talk about two different stories. The first one will be in mammalian cells. I'm going to show you a little bit about how mammalian cells cope with the aberrant accumulation of mitochondrial proteins. And then I'm going to tell you about how cells sense that they have this problem and activate a response. So to start with, we'll talk about mammalian cells now. We wanted to understand what happens to these proteins that are now not imported into the mitochondria. Where do they accumulate in the cell and what do they do? And John Kim, a graduate student from a lab, decided that he wants to tackle this question. So the first thing that John wanted to do is to find a way to look at a protein when it's not imported, some kind of like a reporter for a problem with protein import. And as I told you before, some proteins go into the mitochondria and their MTS is cleaved after the import. And this includes a mammalian protein that's called OTC. So we expect that when OTC is not imported, when it's outside, we'll see a precursor that is bigger and can be separated from the mature protein that is incorporated into mitochondria. So John looked at OTC and he decided to really blast the cells with drugs and inhibit their mitochondrial protein import in a very harsh way, but by adding two ionophores, CCCP and valinomate. So he was very surprised to see that even after 24 hours, we couldn't really see much of this precursor accumulating. So he asked, why is that? And the first thing that came into our minds is that maybe cells are just degrading them very rapidly, and this is why we don't see them. But that was not the case. When John added either a lysosome inhibitor or a proteasomal inhibitor, MG132, he couldn't see any accumulation. So what happens? Where is this precursor going? Why don't we see it? We then came up with the idea that maybe the problem here is not degradation. Maybe we just don't have a translation or an efficient translation of this protein under stress. And indeed, we know that different stresses, including mitochondrial stress, can activate the ISR, the integrated stress response. And this response works by phosphorylating the translation initiation factor, EIF, to alpha, and just attenuate, in general, all translation in the cell, except of specific transcript. We can also block this from happening by adding ISRIP, a drug that inhibits this response. And we indeed saw that when we use ISRIP in these cells that have mitochondrial stress, we all of a sudden see a very nice accumulation of OTC precursor. And this was also true for other proteins like the NDUF6S. So that means that there is some kind of like a response in the cell that prevents from translating proteins that can be harmful. But it also gave us a very good tool to look at these proteins. And we decided to see whether these proteins accumulate in the cytosol or on the mitochondrial surface. So John took these cells and he isolated mitochondria and separated them from the cytosol. And you can see here that we barely, like there is a very faint band if you look at my computer, very faint, small population of the OTC precursor that accumulates in the cytosol, but most of it is in the mitochondria. So that means that it actually finds its way into the mitochondria, but cannot go in, it cannot be cleaved. So then John asked, what if we have a situation of an intermediate where this protein is trying to go in, but it's somehow stuck in the channels on the way in? So if this is true, what he predicted is that under no stress, we'll see a little bit of interaction with this import machinery, TOM 40 and TOM 20, because the protein just transiently go in, but under stress, this interaction will be much stronger. And this is exactly what he saw. He saw both TOM 40 and TOM 20, much more interaction under stress versus control. So that tells us that there is accumulation in this TOM channel. And that is actually interesting because we showed a few years ago that this phenomena of clogging of TOM exists in budding yeast too. So now we know that it's something that happens in mammalian cells too. Now this is a very important point because it's not only that these proteins accumulate somewhere in the cytosol and they need to be degraded, they actually block the way into the mitochondria. So now even if the stress is resolved somehow, we might still have a problem with no possibility to get new protein into the organelle. Okay, so from one hand, I showed you that mitochondrial stress activates this ISR and reduces the translation. From the other hand, I showed you that we have some clogging of the mitochondrial entry channels. Can the cells have mechanisms to unclog these mitochondria? In yeast, we showed in the past that these triple ATPase can be recruited to the mitochondrial TOM and TIM and extract these proteins when they are stalled under stress. The yeast ATPase MSP has an ortholog in mammals, ATAD1, so we decided to look at it. And here, what you can see is that when we pull down ATAD1, we could see that OTC is going with it, so it interacts with it, but only under stress conditions. So again, we have some kind of involvement of the ATAD1 with the engaging with the precursor under stress. And we also were able to show that this interaction is stronger when we express an ATAD mutant that is a trap mutant. This protein does not have the ability to hydrolyze ATP. So it binds to the substrate, but cannot let go. And even though the expression of the wild type and mutant versions is a little bit different, we were able to calculate it and to see that we have an enrichment of binding to the mutant ATAD1. And that means that we have indication here that the OTC is an actual substrate of ATAD1. Okay, so if this is true and ATAD1 deals with these precursors, it should affect the accumulation of them under stress. This is exactly what we were able to show here. When we knocked out ATAD1 from the cells, we see that we have much more accumulation of the OTC precursor, and we also see that we have interference with the little import that you usually have even under this stress. And we could recover it by reintroducing the wild type ATAD, but not the mutant one. Okay, so until now, I showed you a few results, but they were all done with very harsh drugs. And we wanted to know whether this happens also under conditions that are less harsh, that are more even physiological. And to do that, we started looking at different cell lines. And you can see here that most of these cell lines, we don't even see any precursor accumulating, even when we add ISRIP. But we found that in this cell line, which is a fibrosarcoma cell line, we have a little bit of accumulation, and it increases when we add ISRIP. Here, we don't have any effect on the mitochondrial import. We just stop the ISR from being activated. And this is kind of supported by other reports that show that this cell line has mitochondria that are not in the perfect state and they undergo degradation all the time. So if these cells have a higher basal level of ISR, we should see it by looking at the EIF2 alpha. And this is exactly what you see here. We see that the EIF2 alpha is more phosphorylated, which supports that they have some kind of an intrinsic stress that is happening all the time. When we knock out ATAD from these cells, we see that we have more precursor accumulating. But even we see that even if we don't have ISRIP, which means that ATAD1 also here has a role in the elimination of this stalled precursor. But what's interesting is that when we looked at the expression of ATAD1 in these cells, we saw that it's relatively lower than other cells. And this is also seen here by this in online databases. So we thought that maybe if we can express more of these ATPase, we can actually help the cells that are always coping with some kind of mitochondrial stress. And that was indeed the case. When we stably expressed ATAD in these cells, we saw that they have a better proliferation rate. And that was not the same in other cell lines like HEC293s. So we actually were able to show that ATAD can contribute to the health of these cells with mitochondrial problems. So what I showed you until now in this part of the talk is that in mammalian cells, we found that when there is a problem with mitochondrial protein import, proteins tend to accumulate outside, but also in the TOM channel. And when this happens, cells cope with it with two different ways. They generally reduce translation to stop the accumulation of this protein, but they also can directly unclog the TOM by recruiting these AAA ATPase to the TOM channel. Okay, so now I want to move to the second part. And here, again, I'm going to focus more on the transcription activation of a transcriptional stress response. And this is all done in yeast. So the story that I just told you about the clogging of TOM and how we discovered it in yeast, when we did that, we also showed that we could detect a specific stress response, a transcriptional stress response that is activated by protein import stress. We called it the mito-CPR, for mitochondrial compromised protein import response. And what we showed is that the transcription factor PDR3 is a very major regulator of this response. So it activates different genes that are supposedly going to help us with mitochondrial stress. The genes that we saw, some of them actually we still cannot explain why they help this type of stress. They include drug resistance genes and lipid biogenesis and transport and transfer genes. But what we were able to show is that some of these genes that are regulated by PDR3 actually help the unclogging of TOM. And what they do is they help mediate the association of these AAA ATPase with the TOM channel to remove the clogged protein. We also showed that by activating this response, cells can recover better from mitochondrial stress and they have better mitochondrial functions. But the big question that we couldn't answer by then was, how does it happen? How is a stress that is happening in one part of the cell, in the mitochondria, is transmitted to the nucleus to activate this response? And when Christina joined my lab, she's now a PhD student and she's here in the audience, she decided that she wanted to understand how does it happen. And Marina Volpe, a research associate, decided to help and join the project. And the first thing that they did is to look at PDR3 to see if we have any change in the localization of this transcription factor during stress. And you can see here that that's not the case. PDR3 is always in the nucleus. We don't see it near the mitochondria, even when you have stress. And that was also true when we looked at the binding of this transcription factor to the DNA. It seems to not change. So that means that PDR3 is always in the nucleus, and we need something to be delivered to the nucleus to tell PDR3 that there is something wrong. We decided to do a genome-wide screen in yeast to find this factor. And we did it by using an overexpression library of the whole yeast genome with the idea that if we overexpress this factor that activates PDR3, it's going to be enough to activate it even without a stress. And these are the results of the screen. You can see here that all these blue dots are the highest hits that we got. And all the genes that have names next to them are mitochondrial genes, encoding mitochondrial proteins. You can see here in this table that they are really enriched here, which straightened the idea that there is a connection between mitochondria and this response. But what's going on here? Why do we get so many genes? From one hand, it can tell us that maybe something really cool is going on here. Maybe under stress, when mitochondrial proteins are accumulating outside, they can actually interact with this transcription factor and activate it. And usually it won't happen because they'll just be in the mitochondria. But there is one thing that I kind of didn't tell you until now. We showed before that when we overexpress some of the mitochondrial proteins, just by overexpressing them, they go to the mitochondria and just disrupt things there. And they cause stress just because they go to the mitochondria. So we had to somehow distinguish between the scenario that the overexpression of these proteins affect the mitochondria and the scenario which they directly activate the transcription factor. How can we do that? We decided to put a tag in front of the mitochondrial targeting signal of all these proteins. And we know that just by adding this small peptide, it's going to interfere with the entry into the mitochondria. So what do we get there? We get overexpression of proteins that are either going to be in the mitochondria and outside, or overexpression of proteins that cannot ever go inside. They have to stay outside. So this is what you see here. So first of all, this is with no blocking of mitochondria localization. This is just what we got from the screen. And you can see here that we could verify these are some of the hits. They are activating the stress. But when we add this peptide in front of them and block the way into the mitochondria, now most of them cannot do it anymore, which means that indeed most of these hits just caused the mitochondrial stress and didn't really activate PDR3. They activate PDR3 directly. But we were lucky because the strongest hit that we got, MG1, could still do that, even with no ability to get into the mitochondria. So this is where MG1 is in the screen. You can see here that when we overexpress it, it induces the mitochondrial stress, and these graphs that you're going to see a lot from now on are just measuring by qPCR the level of one of the mitochondria of the mito-CPR target genes. You can see here that when we delete PDR3, we abolish this. So this is through the mito-CPR. What we are looking at here is the activation of the mito-CPR. We saw this car outside the LSI the same week that we did the screen, which would probably save us a lot of work if we actually took it seriously. So now you know that you can look around you and look for clues. And in any case, everything pointed to the right direction. Seems like MGE is actually doing something. So what is this protein? What is MG1? MG1 is a co-chaperone. It's a homologue of the GRPL, and it's working with the mitochondrial HSP70 in importing and folding proteins into the mitochondrial lumen. And of course, it has a mitochondrial targeting signal, the same signal that we talked about before that is cleaved when it reaches the mitochondria. When we looked at this overexpressed MG1, what we saw is that it's in the mitochondria to some extent, but it also accumulates in the nucleus. You can see here the DAPI staining. And that is also true for the version that is blocked from getting into the mitochondria. So that's another clue. It actually can go to where the transcription factor is. So we know that the overexpression can do it of MG1. But what does it tell us? Can the endogenous MG1 do it? Can the endogenous one activate PDR3? And also, is it the only mitochondrial protein that does it? Or is it a huge group of many mitochondrial proteins that can do that? To answer this question, we decided to look at the essentiality of MG1 in this process. So we had to find a way to induce protein import stress. And the way that we do it is by overexpressing this protein PSD1 that just overwhelmed the import machinery. So this was just the method. When you do that, you can see that MG1 that accumulates only as the mature protein in normal condition accumulates also as a precursor. So we have an evidence that some of this protein cannot reach the mitochondria during stress. The problem is that MG1 is an essential gene. So we could not just delete it, but what we did do is to add an inducible degrant to its C-terminus. And in this way, we could remove the population that is outside of the nucleus. So basically, when we have import stress, we usually have MG1 accumulating in the mitochondria and in the nucleus. When we induce the degradation, we get rid of only whatever is outside. You can see it here. And the reason is because we don't have proteasome in the mitochondria. So this is a cool way to get rid of this protein only outside of the organ. And when we do that, we don't get any longer an activation of the mito-CPR. So that means that MG1 endogenous is important here, but it's also essential. That's the only mitochondrial protein that does it. So if MG1 activates PDR3, does it bind to PDR3? The answer is yes. Here you can see that Christina looked at a co-IP between PDR3 and MG1. And when she pulled down PDR3, she saw that only under import stress, but not under control conditions, MG1 is interacting with PDR3. And it's not just the MG1, it's the upper band of MG1, the band that is actually outside of the mitochondria. So everything fits here. So it seems like MG1 goes to the nucleus, activates PDR3, but what about MG1 is so special? Of course, for me, and Christina, we already had so many hypotheses about how this protein can act as a co-chaperone in the nucleus and do this and that and work with the cytosolic HSP70. But of course, life is not what you want it to be, and science is complicated. There is nothing to do with the co-chaperone activity. What we were able to show is that the targeting sequence, the N-terminus area of MG1, is responsible for the activation of PDR3. And we saw it here. Calvin Yip from this building helped us to look at this alpha-fold multimer, and we showed that the targeting sequence is predicted to bind to this transcription factor. So is it really true? What we did here is we took this targeting protein, the full-length protein, and we also made a truncated mutation that doesn't have the MTS. And you can see that it completely lost the ability to activate PDR3. And this is, of course, overexpression. What was interesting about it is that this truncated mutation was no longer accumulating in the nucleus. So we weren't sure if the problem with activation was because there is no activation or because that it doesn't go to the right place. So Christina came out with this cool idea of adding an NLS, nuclear localization signal, to this truncated protein, forcing it to go to the nucleus, and then asking whether it can activate the response, and the answer is still no. So this peptide, this N-terminus of MG1, is important for the activation. Okay, so what else can we do? How can we show that it's actually maybe essential? We took the MTS of MG1 and we put it on another protein. This is another matrix mitochondrial, lumen mitochondrial protein, ILV2. What we did here is a chimera where we have the mature ILV2, but the MTS of MG1. And when we overexpress it, it was enough to activate the response. So even on another protein, this small peptide activates the response. And this was even true if we just took this MTS of MG1 and put it, infuse it to a fluorophore, to an M-cherry. That was enough to activate the response. So this is the N-terminus of MG1. This is the MTS, this area. We collaborated with Kevin Wei, also from this building, to look for how this sequence is changing within species. And it's very much not conserved. It's changing all the time, and that is very typical to mitochondrial targeting signals. But what we saw here is that the first 20 or so amino acids are more conserved. This is like in different Saccharomyces. So when we took this area I just showed you and we overexpress it, we see that the mito-CFTR is activated. We decided to look at the other areas. What we were able to show is that when we truncate this area that is conserved, we abolish the response. And on the other end, when we just take these 20 amino acids, it's enough to activate it. So it's even smaller. It's just the real N-terminus of MG1 that has a huge role in mitochondrial protein import, but also here. Okay, so again, the last part that I showed you about everything that I showed you about the MTS was done with overexpression. You are allowed to be skeptical. We're skeptical too. We said, is it really important in the endogenous MG1? Again, MG1 is essential, so we could not just delete it. What we did is to replace its endogenous MTS with another MTS of a protein that doesn't even exist in yeast. And the idea here is that this MTS will have one role: to send the protein into the mitochondria and keep the yeast alive, because once it's in the mitochondria, it's no longer will be part of the protein. But we, in this case, can make a separation of functional allele, where the mitochondrial function is still relevant, it's still happening, but whatever is outside, the MTS role outside, doesn't exist anymore. And when we did that, Christina saw this amazing result that just by switching the MTS of MG1, we could completely abolish the activation of the mito-CPR. And when we use this strain, but reintroduce MG1 to it, the response was back. So it didn't actually lose the ability to activate the mito-CPR. What I showed you until now, again, was activating an import stress by overexpressing a protein, PSD1. We again wanted to see what happens in a more physiological system. So we took yeast that have mutations in their mitochondrial DNA. This is a very good tool because it actually kind of mimics what happens in some of the mitochondrial diseases in humans. And what's nice about these yeasts is that the problems with mitochondrial DNA cause problem with membrane potential and eventually problem with protein import. And what you can see here that indeed, when you look at MG1 in these cells, and this is the endogenous one, you see that it also accumulates outside. So import is not perfect here. We have a good system to look at import stress. And as predicted, in these cells, we have activation of the mito-CPR because, again, they have a constant chronic import stress. But what happens when we remove the MTS from these cells? We again, abolish the ability to activate the mitochondrial stress response. So it's really this peptide that does all the work here. And you can see here that Christina looked at the half-life of this unimported MGE and compared to other mitochondrial proteins that are not imported in these cells with chronic import stress. And she saw that it's also more stable, which is consistent with the fact that it has a role outside of the mitochondria. Okay, so just to summarize the second story that I showed you today, we show that MG1 is a very important and essential co-chaperone in the mitochondria. And under normal condition, it goes to the mitochondria very efficiently, and we never see it outside. But when there is something wrong with the mitochondria and MG1 starts accumulating outside, it can actually be targeted to the nucleus. And there, it's N-terminus peptide, and indeed only the 20 amino acids that are at the first part of its N-terminus can bind to this transcription factor and activate the mitochondrial stress response mito-CPR. Now, this is a very important point because we know about these MTSs for a long time now. And we only know that they have a role in targeting and importing of proteins into the mitochondria. This is the first time that we could show that these peptides act as a signal peptides during stress. So they have another role outside of the organelle. Okay, so with this, I want to thank my lab. I have an amazing group of people. This is again, oh, I forgot to put to show you John. This is John. He did the first story. He was leading the first story that I showed you. Christina and Marina worked on the second part. Tony is an undergrad in the lab that works with Christina now and help her finish the story. This is a cartoon of my lab that we are we really like. And I want to thank also our collaborators, Calvin and Kevin, and our the funding agencies. And thank you so much, all of you, for listening.
Hilla, thank you so much for that. That was really interesting. I've got lots of questions. Let's let's start with maybe some protein chemistry. I'm very interested in all of these interactions that you are starting, I guess, to identify. Is there evidence for direct interactions between these proteins that I guess you're mostly pulling down?
Right, right.
Such as maybe between TOM20 and ATAD1? Is there a direct interaction, or is this part of a mega complex?
It's a great question. The quick answer is that I still don't know. In yeast, we show that the interaction is at least increased when you have a different factor that is expressed only under stress. So only under stress, it goes to the mitochondria and it kind of brings them together. But I still don't know if the interaction is direct. This is something that we still need to look at.
That's great. Can I ask? Okay, go ahead.
Does MG1 have a human homologue?
Yes, MG1 is very conserved from bacteria to humans. We have a little bit of a problem here because the transcription factor that I talked about, PDR3, is not conserved. So what we think is going on is that there is another transcription factor in mammals, but it's not a sequence ortholog, right? It's not similar by sequence, but it's functional ortholog. So this is something that we still need to figure out. But we have preliminary results that show that the mammalian ortholog of MG1 also goes to the nucleus during stress. So we have some hope that maybe it's conserved, but it doesn't also have to be MG1. I think that the whole idea that mitochondrial targeting signals have a role outside the mitochondria is probably conserved, and this is something that we are very, very curious about and trying to figure out.
I have another question. What do you know about mitochondrial stress in viral infection? Does that induce a similar pathway?
In viral infection, it's possible. I don't know of an example off the top of my head that I know that have a problem with protein import, but it is something that it will be very interesting to look at. I agree. Yes.
As you have mentioned before, mitochondrial stress has been observed in neurodegenerative diseases. Has anybody looked at these mechanisms in, say, Alzheimer's disease, Parkinson's disease, and etc.?
Not yet, because it's actually the whole field is quite new, and the idea of clogging TOM was only like we were the first to show it a few years ago. But it's interesting that you're asking because proteins like APP that are involved in Alzheimer's are known to be mistargeted to mitochondria in these diseases. And in the case of APP, it was actually shown that it can clog the TOM channel. So it's going to be very interesting to see if ATAD1 can somehow recover that.
Excuse me. I was wondering, do you know if ATAD1 is the only protein that's capable of unclogging these channels, or do you think that other family members might do a similar role in different cell types?
Right. So are there other mechanisms that are involved in unclogging? So there was another report that showed that P97 or CDC48 is also capable of doing that. It has adapters on the mitochondrial membrane, and it is capable of unclogging TOM. The question that we don't know yet is, are they active in different conditions? Do they have different substrates? These are all open questions that we are trying to work on these days.
Hi, thank you for your wonderful talk. I saw on one of your last slides you had something about low membrane potential. but you didn't touch on it. Could you elaborate what you were inferring there?
Yes. I think you referred to the low membrane potential in yeast. And what we know is that when we have mutations in the mitochondrial DNA that affect the ATP synthase, that's one of the only genes that are found on the mitochondrial DNA. They are all part of the OXPHOS and ATP synthase complexes. And when you have problems with the ATP synthase, you also have problems maintaining membrane potential. And this is a major cause for problem with import stress. I hope I answered your question.
Yeah, thank you so much.
Sure.
Anybody else? Thank you so much.
Thank you.